Every year, foodborne pathogens pose a major threat to public health. Among them, Listeria monocytogenes is one of the most dangerous, causing severe infections with high hospitalization rates. But how do scientists actually track it down in the food we eat? In the United States, the detection of Listeria monocytogenes in food and environmental samples relies heavily on standardized culture and biochemical protocols developed by the Food and Drug Administration (FDA) and U.S. Department of Agriculture (USDA). This laboratory use exercise utilize a modified and simplified version of the standard USDA protocol, adapted specifically for an educational setting. The primary objective of this practical is to detect and identify general Listeria, which is Listeria spp, within the provided samples. However, a key limitation of this modified procedure is that it cannot definitively isolate or confirm the specific pathogen Listeria monocytogene on its own. To achieve a definitive identification of Listeria monocytogenes, supplementary biochemical testing beyond the scope of this initial protocol would be required.
Assalamualaikum warahmatullahi wabarakatuh. So today I will explain about the objective of the Listeria results, lab 5. So, first objective is to detection of Listeria monocytogenes in the food sample. Next is to detect presence of Listeria in food and environmental sample using cultural and biochemical method. Next is to isolate and identify Listeria monocytogenes through selective enrichment and selective media such as MOX and Palcam agar. Next is to perform biochemical conformation tests, including gram staining, catalase test, colony morphology observation, and B hemolysis test. Next is to understand step-by-step procedure involved in laboratory detection of Listeria. Next, to differentiate the Listeria monocytogenes from other microorganisms based on cultural and biochemical rate. So as you can see here, for the result, we got a T-say agar. We got a very even colony growth with a single colony growth for the gain and for the control. So the next one is observation on the microscope using 4 times, 10 times, 40 times and 100 times. As you can see here are the results for that. Next is that one is for gain. Okay, the next one is for the control one. So the control one, as you can see here, it become red. So we will pass on the discussion to discuss about it.
Seringa no beat.
A comparative evaluation between the test samples and the positive control groups demonstrates highly parallel microbiological and cytological outcomes, validating the accuracy of diagnostic workflow. On the primary selective plating medium, both the positive control and the raw meat test samples successfully produced characteristics of colonies wrapped in prominent, diffuse dark halos. This shared phenotypic expression confirms the raw meat sample carried an active, viable load of presumptive Listeria SPP, effectively mirroring the contaminated profile artificially stimulated by the positive control. The metabolic mechanism relies on the enzymatic cleavage of esculin by the bacterial enzyme beta-glucosidase, which releases esculity to react with the ferric ion indicators inside the agar matrix. Furthermore, gram staining of isolates from both sets of plates yielded identical microscopic results under the oil immersion. Purple gram-positive short rod-shaped bacilli, coccobacilli, displaying typical single pad or short-chained arrangements. The consistent performance of both of the differential media indicators and the differential staining reagents across the control and the sample isolates eliminates the possibilities of experimental false positive or technical artifacts. While the positive control confirms that all diagnostic parameters were operating at peak sensitivity, the identical morphology and metabolic profile from the meat sample isolates confirms a genuine contamination event, emphasizing the persistent risk of Listeria collination in raw animal